Welcome back to this course on organic chemistry in biology and drug development. Now, last time we were discussing the determination of sequence in a piece of single stranded DNA. Now, sequence in DNA means the sequence of bases that are arranged one after another. This is extremely important because ultimately whatever is the sequence, that information will dictate what type of protein ultimately will be made in the living system. Okay. So, determination of sequence is the primary requirement in order to predict the following flow of information towards the protein. Okay. Now, I said that there are primarily two methods. One is the chemical method and the other is an enzymatic method. The chemical method was discovered by Maxam and Gilbert, and last time we have a detailed analysis of what is the Maxam-Gilbert sequencing technique. Now, like the proteins, I also told you that if DNAs are very large and it is very difficult to handle such large pieces of DNA, and also it is not possible to determine the sequence of the entire DNA in one go. So, you have to first cleave the DNA into smaller pieces, and these smaller pieces can be obtained by use of restriction enzymes, a particular class of enzymes which are called endonucleases. They recognize typical palindromic sequence within the DNA, and accordingly they cut the DNA. There are two types of cuts, there are blunt cuts and there are sticky cuts. Okay. So, basically what you have to do, you have to take the DNA first, that is the first requirement for any sequencing technique. First you have to cleave into smaller manageable pieces, that is by restriction enzymes, and this was the gel pattern that you get in case of Maxam Gilbert. I just remind you that some books have written that there are 4 lanes, one is A specific lane, another is G, another is C, another is C plus T, but in practice what happens is A and G, there are some few G some bands which correspond to the cleavage of the G can also take place. That is why I said you a G specific reaction and A plus G specific reaction, C specific reaction and C plus T specific reactions, but that serves the purpose. Okay. Now, we will go to the other technique, which is the Sanger's, which was discovered by Sanger's, Frederick Sanger at Cambridge University, England, and that was done in 1975. At the same time, Maxam Gilbert sequencing technique was also discovered. Now, this method, Sanger's method, it is an enzymatic method, but it is also known as a chain terminator method or a dideoxy method. So, these are the other names given to Sanger's method of determining the sequence of bases in a DNA. Now, what is the principle behind Sanger's method? The principle is that whenever there is an enzyme called DNA polymerase. Now, that DNA polymerase job is to make the DNA polymer. If there is a strand of DNA, then what you do, actually what is done in Sanger's method, that if you have a single strand of DNA 5 prime, suppose this is 3 prime, then the DNA polymerase, if there is one strand, then the DNA polymerase can make the complementary strand of this primary DNA strand. Suppose we have this DNA strand and we want to know the base sequence in this strand. So, we have to use the DNA polymerase in order to make the complementary DNA, but there are certain important points that one needs to consider. One is that DNA polymerase works only when there is a small piece of DNA which is attached to this primary piece of DNA. Primary means for which you are trying to determine the sequence. So, a small piece of DNA needs to be attached, and then only DNA polymerase can start from there and make the remaining part of the chain. This piece is called the primer. Now, this is made up of deoxyoligonucleotide. So, this is a DNA primer. So, DNA polymerase, unless this primer is there, DNA polymerase cannot work. So, basically there must be a small segment of double stranded DNA, and then the remaining part can be joined together one after another by the DNA polymerase. So, in Sanger's method, first is that what you need is DNA polymerase, and along with DNA polymerase you also need a primer. Okay. And then the third thing is that in order to make this DNA strand, what you also need is the building blocks for the DNA synthesis. Like if you want to make a peptide, you need the building blocks, those are the amino acids. So, here also the DNA will need the building blocks. Now, what are those building blocks? They are the deoxynucleotide triphosphate. So, basically I can write the structure. So, what you need is this thing, a base, the OH and triphosphate, that means P double bond O, O minus, then O, another P is the same thing, double bond O, O minus, and then again O, P double bond O, O minus. I think I just write on the top of it, that is O minus. So, this is what is nucleoside triphosphate, NTP, deoxynucleoside triphosphate. Okay. Because this up to this point is what is known as nucleoside. So, it is called nucleoside triphosphate. N means it stands for all the 4 bases A, G, C and T. So, basically what you need is all the 4 deoxynucleoside triphosphate. Okay. And why do you need this? Because in the synthesis, during the synthesis of the chain, what happens is that actually it is written here, but I can explain that you have a triphosphate, nucleoside triphosphate. Suppose you have already made part of the, so you have already suppose made the part of the DNA and a 3' OH is free. Now, if you add a nucleoside triphosphate, then DNA polymerase comes, and first let me write phosphate. You can always write like this circle, it is a triphosphate, and then this one. So, DNA polymerase what it does? It attacks this phosphate in presence of magnesium. A magnesium ion as a cofactor is needed to neutralize the charges of the phosphate ion, because the phosphate is negatively charged and you are doing a nucleophilic attack. So, someone has to neutralize these charges, and that is taken care by the magnesium ions. So, the OH attacks the phosphate and an inorganic diphosphate or a pyrophosphate that goes out. So, in the process what you do, you have made you have extended the DNA chain by one nucleotide. Okay. So, this was the earlier DNA part. So, then this again attacks another triphosphate, and that is how the DNA chain is extended. So, this is the principle. Now, some important again points we need to consider, that is the 3' OH, that when the DNA chain is made, it is the 3' OH attacking the 5 prime triphosphate. That is very important. Now, on the top side, ultimately you have a DNA, you have a oligonucleotide here, where we have a nucleotide here, where the 5 prime wedge will be free. And as you go down, finally, when it is truncated, when it achieves the desired length, so your 3 prime wedge will be free. So, the direction of synthesis is from 5 prime to 3 prime end, but that means that it is the 3 prime wedge that is attacking the 5 prime triphosphate. Okay. So, this is the job of DNA polymerase. So, the important thing is that it is the direction of the synthesis takes place from 5 prime to 3 prime, and while doing so it is the 3 prime wedge attacking the 5 prime triphosphate. Now, what Sanger did? Sanger thought that if suppose if one of these deoxynucleoside triphosphate, if this wedge is not there, suppose this wedge is not there. So, this wedge is not there, then what will happen? Then if this is incorporated in the chain, then the chain will stop here, will truncate, because that nucleophilic wedge is absent here. So, during the growth of the, so you have the primary DNA strand and you have the primer up to this point suppose. So, that is your 3 prime. So, the synthesis will not take place from here, because already I have said that the synthesis takes place from the 5 prime to the 3 prime direction. So, if this is your 5 prime going from 5 prime to 3 prime, then your primer should be here, and the primer 5 prime should be here and 3 prime here. Okay. So, that is the primer, and then as you add all these deoxynucleoside triphosphate, depending on the sequence here, whether A is there or G or all these sequences, the complementary base containing nucleoside triphosphate will be brought here, and this 3 prime which is free. So, that will now react with the nucleoside triphosphate and then extend the chain according to the base, whatever base sequence here. Suppose there is the, after the primer the first base is E. So, definitely it will require a GTP, a deoxy GTP, DGTP, that will be brought. The 3 prime wedge will attack the 5 prime triphosphate, and in the process a phosphodiester bond will be formed between the primer and the G. Suppose the next one is T. So, now there will be a DATP that will be brought, and now the 3 prime wedge of the GTP which was used earlier. So, now it is only the G, because the diphosphate is already out, and the 3 prime wedge will attack this triphosphate of ATP. So, that is how A will be incorporated here. Okay. I hope this is clear. So, that means it goes one after another, but the important thing is always the 3 prime wedge is attacking the 5 prime triphosphate. Now, Sanger cleverly did one thing, that he, instead of he took all these deoxy triphosphates, that is all right, that will increase the chain, but along with that he had put the dideoxy. I said this is the dideoxy. So, he also added the dideoxy in TP, dideoxynucleoside triphosphate. And what will happen, that when there is requirement of suppose A, and if there is deoxy, always there will be DATP is there, because you have to add that to increase the chain. But if instead of DATP, if DDATP is taken up by the enzyme by mistake, then what will happen? The chain will truncate at that position. The chain will not proceed any further, chain growth. Okay. If it is the correct, the deoxy ATP is taken, then the chain will progress, the chain growth will progress, but the chain growth will terminate if a dideoxy nucleoside triphosphate is taken instead of the normal deoxy ones. That is why this method is also is called the chain termination method by Sanger. Now, let us take an example, then that will be that will make it more clear. Let us see. So, suppose this is the DNA I want to sequence, and suppose this is the one which I need to know the base sequence. This part is what is called the primer. So, when I have the DNA, it goes up to that point, that means I know the sequence up to this part, I know the sequence. So, accordingly I synthesize, there are ways of synthesis of oligonucleotides. So, I will synthesize the oligonucleotide which is complementary to this part of the DNA. So, what will happen? The primer will now hybridize, will form a duplex here by the hydrogen bonds, and in addition this primer is such that the 5 prime OH, like the Maxam Gilbert sequence, the 5 prime OH is having a radioactive phosphorus. It is tagged with a radioactive phosphate. Okay. The phosphorus is radioactive. That is needed because ultimately you have to find pieces where that label has to be there, otherwise it will be all mixed up with the other nucleotide whatever is present there. So, you need something, a tag which will ultimately act to determine whatever you want to know, to determine basically the nucleotides that are basically truncated. You may not understand right now, once I give the example it will be very clear. This radioactivity is needed to know which nucleotides are truncated by taking the dideoxy nucleoside triphosphates, if that is taken. Okay. Now, let us, so this is my primer. So, one important issue of Sanger sequencing is that if I have the DNA, I need to know the sequence of few bases at the end. Okay. Then I can design the primer, and the primer goes here and binds. This is my primer. Now, what will happen? Suppose this is the, let us forget about this one, let us take a new example. Suppose this is the sequence that this DNA has. Okay. TTAG random, just I am writing just random some sequences. Okay. Now, this, remember this primer starts from 5 prime to 3 prime, that has to be because the DNA sequence goes from the 5 prime to 3 prime direction. So, now what will happen? So, whenever there is A, so it should take a T. Now, suppose instead of T, the enzyme picks up a dideoxy T, then what will happen? This piece will not grow any further if it is dideoxy T. So, let us make some kind of notation. Suppose this is dot, dot means dideoxy T. So, if dideoxy T is taken, the whole thing will stop here. Now, if the correct T is taken, that means the deoxy T, then what will happen? It will continue up to which point? It will continue up to the point when there is requirement of another T. So, C, then G, then A, then A, and now you again come to this point that there is a requirement of a T. So, if the enzyme now takes the dideoxy T, so again you will have a truncated chain here. So, you can have 2 truncated chains, you can have more also if there are more A's, because it depends on the number of A's in the chain. So, you will have others also. So, T, C, G, A, T, because when this is taken, when the correct T is taken, this is going to proceed forward. Okay. So, then C, because it is G, then a G, then A, no problem. Now, then there is a T. So, there is a chance of truncation. Okay. So, it takes the dideoxythymine and that will truncate here. And but if it takes the right one, then you will get another one, T C G A T C G A T. Suppose this is the correct T that the enzyme has taken, but the next A, again there is a possibility that it may take a wrong T. Wrong T means the dideoxy T. So, in a test tube, suppose what I do, I take the DNA single piece, then I add it the DNA polymerase, I add magnesium, I add all the NTPs, deoxy NTPs, the normal ones, then I add in this test tube only DD TTP, that means the dideoxy TTP. Other dideoxy I do not add to this test tube or the Eppendorf. Okay. And what else? I need to add the primer, that is the radioactive primer. Okay. So, the primer goes and binds, and because I have only DDTTP, so the chain will terminate only when there is requirement of T here. Others will not terminate, because I do not have added the DDCTP or DDGTP or DDATP. Okay. So, that has to be made clear. So, if you add this DDATP in one of the test tube, one of the Eppendorf, then you will get 4 pieces which are all truncated at T. Okay. So, if that is clear, now the other part will be very clear. This is what is shown here. These are the Eppendorfs. So, in one Eppendorf, as I said, you add all the 4 D NTPs plus the polymerase, magnesium, everything, primer. But in addition, what you add is DD ATP. So, if you add DD ATP, then the chain will terminate whenever there is a requirement of A. There is a possibility of termination. I am not saying that it will always terminate. Some will take the correct A and that will proceed forward, but some where there is a where the enzyme picks up the wrong A, that means the dideoxy, then the chain will terminate. So, there are 4 now Eppendorfs. In one I have added, what is the difference between the contents here, that in one I added DDATP, in the other I added DDCTP, in the third one I added DDGTP, and in the fourth one I added DDTTP. Okay. So, again I take another example. Suppose the DNA has this type of sequence A G C T, T C A T, G C A, just to make it little bit shorter. So, that because of the because the principle is most important. So, take a short one. Now, my primer suppose is this T C G A. This is the length of my primer. Usually primer lengths are longer than this, but for the sake of the problem I am just taking this part. Say up to this point is the again the direction I have to change. This is the 3 prime and this is the 5 prime, because the primer has to start from 5 prime and then it should go to the 3 prime. Let me just erase write the correct ones. Okay. So, what happens here? The primers are, this is 3 prime direction, 3 prime to 5 prime direction. I have written that sequence, and so that will be 5 prime and this will be 3 prime. Okay. So, up to this point is my primer. Now, what will happen in the first test tube? What I will have? I will have because I have added DDA. So, I will get a truncated chain like this, and then I will get 5 prime T C G A. This is the primer, this is the primer, and then the important thing. So, if the proper A is picked up, that means the deoxy ones, then it will proceed further G. Now, this test tube does not have any GGA, GG-ATP. So, G will always be the right one, that will be picked up. Then T, that is okay. Then there is another requirement of A. So, it may terminate here. Okay. And finally, the 5 prime TCGA, that is the primer, then AGTA. So, you just see whether there is requirement of A anywhere else. So, up to this point is okay, and then C, then G, then T, and then there is a termination. Okay. So, I will get 3 pieces in case of the first test tube where I have added A. So, in this way I can predict without writing that if you added DDCTP, so how many pieces you will get. So, you will see that where are the G's, at that point C will be taken up. So, if you add the DDCTP, so you will get only, there is, I could see only one G after the primer. So, only when here it is, it will be taken up. So, for this one I will get 5 prime, then I just write primer, I get primer is always there, then I should get AGTC and then AGTA, AG again AGTA, and then there is a requirement of C. So, it will truncate here. That is the only piece that you will get from C. So, from DDGTP test tube, how many pieces you will get? You will see the number of G's after the primer, number of C's after the primer. So, you will get here there is a C and there is a C here. Okay. And for TTP, you will get wherever there are A's, then there is a question of truncation. So, you will get here there is a there can be truncated because there is a requirement of T here and there is a requirement of T here. Now, what you do, if you count these numbers, see for this for the first test tube where you have added DDATP. So, you can get nucleotides, oligonucleotides, which is primer plus 1. So, P plus 1 here, primer plus only one oligonucleotide is there. Also, you will get primer plus 4, P plus 4, and you will get in the another one, you will get primer plus 1, 2, 3, 4, 5, 6, 7, 8. So, primer plus 8. So, 8 nucleotides are joined. In the case of C, you will get primer plus 5, I could see. Okay. So, this is your A specific reaction, this is C specific reaction, then your G specific reaction, you will get primer plus, where are the G is required. So, there is requirement of G here, there is C here. So, you will get primer plus 2, and then there is another requirement of G here. So, that will be 1, 2, 3, 4, 5, 6. So, you will get primer plus 6, and for the T specific reaction, you will get primer plus wherever there is requirement of T. So, there is a requirement of T here, that is primer plus 3, and you will get another A, that is 1, 2, 3, 4, 5, 6, 7. So, you will get primer plus 7. That means, all these test tubes are giving oligonucleotides which are varying in length. This is one, then the second one in molecular weight, if we go from decreasing to the increasing. So, here it is the least molecular weight piece, then the second one is in the G lane, and the third one is here in the T lane, the fourth one is here, fifth one is here, sixth, seventh, okay, and then eighth. So, now what we do? We do a gel electrophoresis. Okay. So, now we take a gel electrophoresis and we run 4 lanes. Okay. So, one is A, another is C, another is G, and the third is T. So, what will happen? In the A lane you will have 3 pieces. So, one will be the minimum molecular weight. Then you have somewhere here that is the p plus 4 and somewhere here that is the p plus 8. Okay. So, and then for the C lane you will get only one piece and that will be here, because that will be little bit below than the p plus 4. Then for the G 1 you will get somewhere here that is p plus 2 and then p plus 6 which is somewhere here, and for the T lane you will get p plus 3. So, that will be here and then p plus 7. So, p plus 7 will be little bit here, because this is your p plus 8. So, now you read it from the bottom to the top. So, what will happen first is your A, then A comes the G, then comes T, then comes A, then comes your C. Okay. So, that way you just read it from this to that side. So, what is this A, G, T, A, C? Now, you write, you just check that what is the sequence of the complementary strand of the DNA. So, that will be A G, then A G T, see A G T, and then there is A, then there is C, and then there is G. If you just continue, you will get that. So, that will be your 5 prime end and this will be your 3 prime end. So, basically you read the gel. It is very simple, reading the gel is not a problem. You just see what are the bands, which is the fastest moving, that is the first nucleo, that is the first base, then the second, then the third, then the fourth, then the fifth and sixth, seventh and so on. One important issue is here that whatever sequence you get is the sequence is a complementary, your sequence of the complementary strand to your primary sequence. Okay. So, the actual sequence you just write the actual sequence. So, that will be G, then T, that is not a problem. You can write the, if you get the complementary sequence, you can write the actual sequence like this. Okay. So, that is the basic principle of Sanger's method. So, it is called dideoxy, because the whole thing is based on the dideoxy approach, and the important issue is that finally, at the end of the day you get the sequence of the complementary strand. From that you can write, you have to write the sequence of the primary strand, that is what is you are asking for, the sequence. Okay. I hope this is clear. This is a very intelligent way of doing the sequencing, because this Sanger's method of dideoxy, using dideoxy nucleoside ultimately give the way, ultimately showed the way how to make antiviral compounds. That we will discuss when we go to the medicinal chemistry part, but many of the antiviral drugs that we have today are literally based on this principle of using the dideoxy nucleoside. Okay. So, that means nucleoside which lacks the your 3 prime wedge. Instead of which you can have other groups which is not a nucleophile, that is also possible, but in Sanger's case he used dideoxy, he just do not bother to put any other group, he removed the wedge also. Okay. So, that is what is Sanger's method. So, ultimately you get the this type of gel and you read from here, but as I told you, this is the complementary strand, and finally you can write the actual sequence of your primary strand. So, that is all about Sanger sequencing. Now, in the next session we will first start with what is the problem, what are the problems of Sanger sequencing, because that is how science is developed, that consider the problems, and as such there is no problem, but there are constrictions which are present. So, we will discuss that in the next session. Thank you.